Please let me introduce Professor Jurgen Kuball from the University of Utrecht. Jurgen will review the diversity and subsets of gamma delta T cell lineages and their biology and their potential clinical use. And I'll leave that to you, Jurgen, please.
Yeah, thank you very much. So can you briefly confirm that you see the slides?
Yeah, we can see the slides in presentation.
Yeah, it's for us a lot of genetic engineering, and of course there is no better long-term introduction than Danielle talking about BTNs. But I would at least like to give some slides to show the different flavor. And of course, we all show the slide also he showed on gamma delta T cells, which associate with a positive clinical outcome. And then we say this is even better than NK cells. And of course, there are multiple layers of cancer immune surveillance where gamma delta T cells are important. However, if you carefully read these papers, there are also a lot of tumors who don't have tumor-infiltrating gamma delta T cells. And that's what all genetic engineering is about, because you try to add an immune repertoire to a patient who doesn't have the appropriate immune repertoire, and we try to harvest the feature of a gamma delta T cell for that. And I think another thing frequently mentioned is also that because of the poor understanding, all the clinical trials that are out so far have been mainly negative with anecdotal clinical responses. So we thought it's a lot about homing properties. We heard about this. It's about proliferation deficiency of gamma delta T cells, and I will dive into the completely underestimated diversity of gamma delta T cells, in particular also of gamma 9 delta 2 T cells. If you look at the two different flavors, of course, the first one I will dive in is the gamma 9 delta 2 T cells, where we together with others described the different parts of mode of action. The first one is, of course, as Daniel extensively described on the BTN3A1. And later on, there was coming up the BTN2A1 story out this last year, which I think were convincingly shown that this interacts with the gamma chain of the gamma delta T cell receptor. And as Daniel also pointed out, there is CD45 suggested as a potential ligand for BTN3A1, where that's really true. We are not sure. And I want to highlight our contribution to the fields on the right side. On the one hand, we had more than now 10 years ago, and also in this year, a paper really highlighting the importance of the variations in the CDR3 region of the delta chain, which in particular, if you engineer cells with a gamma delta T cell receptor, but also as we published just now, do bispecific formats, the sequence of the CDR3 region is really either the success or the failure of the strategy. And another important one we published last year from Anna Viberova is the fact that there is a huge dynamic also in BTN3A1, and maybe one of the reasons why Danielle doesn't see so much toxicity is because the dynamic of this molecule is differently regulated in healthy and tumor cells by, for example, ROB, but most likely also other players. If you start then cloning gamma 9 delta 2 T cells from healthy individuals, that's what we published here. I was really surprised about the functional diversity of all these cells, because you see some cells here clones in blue, which have a high frequency and are very tumor reactive, but you have also a high frequency of poorly active gamma 9 delta 2 T cells in the blood of every healthy individual, meaning if you would like to translate this for example, as a third-party product into a patient, you will frequently run into the problem that you have also high frequency of cells in there which are poorly active. And of course, at the end of the day, you would like to give, as always in medicine, the most highly compound to your patient and get rid of the non-active compounds. That's why we started working on the TEG concept, where we say we just will sort of piggyback on the CAR-T concept and use CD4 and CD8 cells as a carrier and introduce a gamma 9 delta 2 T cell receptor, which we carefully selected from all the receptors we cloned, the most highly active one against cancer cells. And we thought we achieved two different goals by this. On the one hand, we uncouple this gamma 9 delta 2 T cell receptor from a lot of innate immune receptors, just papers out also that, for example, NK-G2A might have a negative regulatory impact on that. On the other hand, you reprogram CD4 and CD8. So you introduce in the concept of a gamma 9 delta 2 T cell receptor the idea of help. which has been shown in all different immunotherapies as key for their success. We started based on this. This is an academic clinical trial. A long journey. In 2011, we published the first receptor. Then we optimized the receptor. We had a couple of reports on the mode of actions. We looked also at efficacy and safety in 3D bone marrow models, also led by Jean-Sebastian. And of course, 3D Stratemans put a lot of efforts in the engineering strategy, which is rather complex in the field of ATMP, but we made it to the clinic and are currently in the dose escalation of the clinical trial. If it works, yeah. I want to highlight one interesting aspect on the engineering strategy, because if you engineer the TEGs, how we call them, you see that they downregulate the alpha beta T cell receptor, and we use this actually by using, and I think this slide was skipped from a certain transplant strategy to delete non-engineered or poorly engineered alpha beta T cells. And we thought it might be a good idea also to use maybe such an antibody for other engineering strategies. And although this is all about gamma delta here, I think alpha beta is also quite interesting. So make a sort of sidestep into this, where we took advantage of the fact that murine and human alpha beta TCRs are not cross-reactive with a particular antibody, so we could really do epitope mapping on where is this depletion antibody binding, and found out that only a couple of amino acids are important for this. So you could engineer an alpha beta T cell receptor with certain mutations, which does not allow this GMP grade anti-alpha beta TCR to bind. And then you see on the left, that's the engineered product without mutations. In the middle, you see when we introduce a mutation, you see a downregulation of human alpha beta T cell receptors. And on the right, you see again, then the purified fraction. So meaning this is an elegant strategy, originally inspired by the gamma delta T cell field, also to make purely alpha beta T cell engineered cells. And we showed also, if you have time to read this paper, that this particular strategy you can also use to engineer antibodies which are binding these mutated sites. So at the end of the day, you could have also a kill strategy, how you can eliminate engineered immune cells. And in particular in the field of alpha beta T cell receptors, that's interesting because they target also potential self-antigens. But coming back to gamma delta T cells and gamma delta T cell receptors, of course, the main reason why we got also interested in this, because it's not only interesting for liquid tumors, but also for solid tumors. And I may draw your attention to this in progress publication, which is close to resubmission, where we use TEGs in breast cancer. And this was an excellent collaboration, actually initiated by Florian Deckers and Sebastian together, where they took advantage on the one hand on our TEGs, on the other hand, through Hans Klevers, we had access to breast cancer organoids, and then we tested the recognition of these organoids by our TEGs, by different techniques. We looked in single cell sequencing at their behavior to better understand what are active fraction and non-active fraction. And just to give you some highlights, and again, hopefully soon completely online, this is just a snapshot through the different breast cancer tumor cell organoids we had from different tumors. And as you can see, different flavors, HER2 new positive, ER, PR positive or negative. And you see that some are recognized and non-recognized, and that doesn't depend on the hormone receptor status. But if you then look at the properties of these tumor cells, mainly tumor cells which have a strong interferon signaling machinery have been recognized. While if they are not present, they have been poorly recognized. Even more interesting is then, because with this different elegant technique, you can really follow hundreds of cells in this microscope and see how they're moving. And as in life, as maybe also now behind the screens, you have the static ones who are just sitting behind the computer, but you have also super engagers who are looking in the left and the right and maybe twittering and looking at LinkedIn what's happening. And of course, you would like to have in your product a lot of super engagers, maybe some ticklers, maybe some super scanners, and you would want to understand the properties of the different cells. So if you engineer a product with such a gamma delta T cell receptor, are we able maybe to even increase efficacy by getting us the subfraction? And most likely, these properties are also not so different if you think about CAR T engineering. So I think it has an impact on both on gamma delta TCR engineering and CAR T engineering. And I hope this will work now here, because this is really a very nice movie made by Schultz and Florine, where you see in the blue one, it's TEG, and the yellow one is breast cancer organoid cluster. If something is red, is dead. And as you can see here, these cells, or this cell is moving there, it's sort of attaching to the tumor, and then it gets widespread arms and starts really to bite around and really is eating up this big cluster. And you will not be surprised by single cell sequencing when we asked what are the different properties of these cells, and we could identify a lot of new genes which are important for morphological plasticity of these cells, which is not surprising if you see this, because this seems to be a key feature to become a super engager. You need to be super in terms of plasticity to be able to really bite down to the end of the tumor cell. So I think very important to further improve also products. I can also show another example where we have seen that NCAM1 apparently is in particular overexpressed in the super engager. So again, making the next generation of products would maybe mean that if you engineer these tags, you are looking for NCAM1 positive cells, which you highly would like to infuse into the patients, because these are the highly active ones. One further down, even in this direction, is then if you look at the machinery in such a T cell, you can see that there are certain waves of signaling pathways. And the one that was picked out by them was interferon beta, where we saw that there might be a deficiency in interferon beta signaling. And if you add this then to tumor cells, which should be recognized by TEGs but are not, you can see that you can turn non-recognized into recognized breast cancer organoids. So again, a very valuable technique to really understand on a molecular level what is happening if a T cell is entering the tumor tissue, and how can you further improve the therapy. This works also in vivo, also only a snapshot of the experiments we did, where you can now with true tumor organoids, breast cancer infused into mice, you can see that these TEGs are able to control solid tumor outgrowth. Bringing us to another class of gamma delta T cell receptors, because it's not only about gamma 9 delta 2 T cell receptors, we have also a lot of delta 2 negative gamma delta T cells. And I think there, the understanding of the receptor ligand interaction is even, still way much more work than for the gamma 9 delta 2. And I just want to highlight 2 or 3 papers on this. The one is we did together with Prof, where he really punched out gamma delta T cells from the tumor tissues. We sequenced the TCRs and found that there's a high tumor activity in the TCRs we punched out. But again, it also highlighted that not all the tumors have really tumor-infiltrating gamma delta T cells. So again, another reason as to why engineering strategies might be interesting. There's still, in particular for the delta 2 negative one, the debate ongoing, is it really the T cell receptor which is so important? Or, as Adrian Hayday would say, isn't it only the NK-receptor driven killing of the tumor cell? I think still an open debate. What we for sure found was something actually that we rediscovered, because initially, if you read all the reviews, also we have been writing gamma delta T cells should not be able to recognize classical HLA. But we found in our hunt for new gamma 9 for gamma delta T cell receptors, also a delta 2 negative gamma delta T cell receptor, which was able to recognize HLA-24. And this is the series of papers where we published this. And I just want to highlight one experiment, because from our Japanese colleagues, we got HLA-A24 transgenic mice, because this is what this gamma delta T cell receptor recognized. And now, to our biggest pleasure and surprise, this receptor efficiently eliminated HLA-24 tumors, but it did not, as you see here, if we just infuse the cells into these mice, did not have any toxicity in the mice. I'm not sure whether it's popping up. So it should pop up here. Here it is. And these mice are just surviving. So an interesting twist where apparently HLA-24 is recognized on tumor cells, but not on healthy cells. We think it might have to do something with clustering, but all details are not figured out so far. Another important feature to understand if you look in these delta-2 negative gamma delta T cell receptor, in particular this particular gamma 9 delta 1 T cell receptor, was that this was cloned from an CD8 positive clone. So, and we quickly figured out that this gamma 9 delta, that this gamma 5 delta 1 T cell receptor is CD8 dependent. So that would mean that in the engineering strategy we used so far, engineering CD4 and CD8 positive T cells, only CD8 positive T cells would be redirected. And of course, we know from many engineering strategies that might be suboptimal, and you might also want to reprogram CD4 cells. This we did, and it worked, to put it in a nutshell, as you can in particular see on the right side. So on the one hand, you have TEGs 011, we called them, which don't have CD8 alpha co-expressed, and you have then TEGs 11 CD8 alpha co-expressed, where you see that they clearly can better control the tumor in vivo. And on the left side, you see the formal proof that, of course, CD8 is able to reprogram then CD4 positive T cells. So keep in mind that in particular, I think, although this sort of is an interesting twist of nature, the HLA reactive one, which also other groups reported most recently, but I think these receptors might more heavily depend on co-receptors than the gamma 9 delta 2 T cell receptor. Bringing me to a last part. Of course, I'm really originally coming from the field of cellular immunotherapies. Totally believer in this, because I think you can really implement for a long-term an immune repertoire into the patient, in particular if you're engineered in CD4 and CD8 positive T cells. But of course, it's also quite a cumbersome and time-consuming strategy. So that's why we thought a while ago, why not making bispecific formats where you have soluble extracellular gamma chains, soluble extra delta chains, you have a flexible linker, and you link this to an anti-CD3 SCV, and this should then optimally connect the target cell and the T cell. So to put it in sort of in this nice graph, you have phosphogen antigen overexpression, which in our understanding is not presented, but it's in an inside-out signaling, then activating BTN3A1, BTN2A1, and this is connecting then the tumor cell to the T cell, and both together is resulting at the end of the day in a T cell activation, and on the other hand, cytokine secretion and tumor killing. I think reasons why this should not work are manifold, and Paul asked me this a while ago, so I really looked for the numbers, because if we look at alpha beta TCR affinity in CD4 and CD8 positive T cells, they range between 40 and 70 micromolar. And if you think about the published alpha beta TCR-based bispecifics, there was it even necessary to make an affinity maturation to the picomolar or femtomolar range to make this happen. So, and then if you look at the publications about the gamma delta T cell or gamma 9 delta 2 interaction with the ligand, of course, formally you can also only do it with the gamma 9 chain with a BTN2A1. It's about 45 to 50 micromolars. So this format should not or only poorly work. But of course, this is only half side of the game. We thought also at this moment, because you have also a delta chain, maybe this can contribute. And furthermore, of course, in the alpha beta TCR engineering, you're hunting for some peptides presented in MHC molecule, and maybe this BTN3A1 BTN2A1 interaction is so much overexpressed in there that also lower affinity might be suitable for this engineering concept. This is again sort of the data we just put out. And it's really amazing that also the original clone we worked on, the clone 5, and another one we published the last paper, the A3, they've by far the best clones, nearly a log better than all the other receptors we hunted out. And we really went also through all the other published gamma delta T cell receptors. I may proudly say none of the receptors is as good as the 2 receptors sort of we found so far. In particular, in this concept, you really also not only have a powerful engineered text, but you have also a powerful GAB format. This is a formal proof in a very elegant model, in a 3D model, where on the one hand you see if you add GABs to the 3D model, they really start inducing T-cell infiltration. A little bit what Danielle showed on the tumor. We also in this model are able to look at stroma. Stroma is not targeted, and on the other hand, the different tumor cells are targeted. Oh, this is going quicker than I thought. So you see different tumors are targeted. So proof of principle that the SCAP format might be a very quick way, like we use, for example, also CD19, CD3 bispecific antibodies in the clinic to really to attack a higher tumor load. This is the formal in vivo experiment. Of course, we just started this one because setting up these bispecific tumor models is quite a challenge. In this model, we only injected them for the first, from day 9 to day 15. Of course, you can look at all different ways how you inject this, and of course, they are washed out over time. But at least the reviewer said that's fair enough for this model to see some tumor control. And I think there's really great potential in this type of engineering. So bringing all these data together, I showed you functional diversity is really very important, and that's why I really believe in strategies where we purify TEGs and engineer TEGs. I made this quick sidestep to the alphabetic engineering. Keep this in mind if you in the future might work on this. We have GMP-grade antibodies and engineering strategies which allow to purify also alpha-beta TCR engineered cells. I showed you that we are, with the TEG concept, really ready to go to move from liquid to solid tumors. Breast cancer is the model I showed, but we're pretty sure others are also recognized. Colon, I think we showed also in a couple of reports the last time. And Behave is a very powerful strategy to really to understand your product and to further purify it and improve it. I showed you this interesting twist of delta-2 negative gamma delta T-cell receptors, which recognize tumor HLA, I call it, with the help of CD8. And at last but not least, the new kids on the blocks are for us the bispecific concepts, because they're in a very rapid way you make sort of a third-party engineering, and you can, because alpha beta T cell is a T cell in all cancer patients, which is the last cell to survive, you can maybe easily connect then the alpha beta T cells to the tumor cell. And I showed also that these gaps induce infiltration, at least in the models. At last but not least, this is really group effort, and really shout out to Charles Sebastian, who did a lot of this work, Trudy Stratemans, a lot of the ATMP production. Dennis Beringer is really the bispecific god at our lab, together with a lot of enthusiastic PhDs working there and technicians. Thank you so much for that. I should have one more slide. Oh, this disappeared apparently, where I think a lot of collaborators, because this is, of course, in the gamma delta T cell community, otherwise not possible. And of course, as you've seen, Hans Klevers, Florian Deckers, Anne Rios have been essential for this solid tumor models. Thank you very much for your attention.